Now we're going to look at DNA profiling, which is a pattern produced by an X-ray film. So all individuals except twins have a unique DNA profile. So let's look at the uses of a DNA profile. One of them is to identify crime suspects. Number two, we use DNA profiles to prove paternity. And another reason is we use it to determine, let me write it properly, to determine the probability of genetic defects. Another reason for the use of DNA profiles, it is to establish the compatibilities of what? Of tissue types. For organic transplants, for organ transplants. And the last use is to identify relatives. So these are the 5 uses of DNA profile. And one other thing is that when you use the DNA profiling, the interpretation and comparison of DNA profile must be held with caution. The reason for this is, reason for caution in DNA profiling. Okay, the first reason for caution in DNA profiling, the first one is humans interpret the results, which means mistakes could be made. This is one of the reasons why we need to be cautious of DNA profiling, is that humans are the ones that are interpreting the results. And the second reason is that the method of profiling may be different in different laboratories. And the third reason may be that only a small piece of DNA is used in profiling, so the profile might not be 100% unique to a particular individual. The DNA profile is expensive and therefore not readily accessible to those who cannot afford it, particularly in crime cases. And the last reason why it is, DNA profiles may reveal information about a person which could be used against them in a prejudicial way. So these are the reasons why we need to be cautious with DNA profile. Okay, so what we have here is a diagram which shows us what happens inside a nucleus. If you remember in our previous videos, we said that DNA is found inside the nucleus. So what's happening here is what we call protein synthesis, which is the formation of protein, right? So a protein is made up of amino acids, right? So which you can simply call the monomer, because they're building blocks of proteins. So this is our DNA here, which is double helix. It is inside our nucleus, and so due to the presence of enzymes inside this nucleus here. They're going to break the bond of the DNA, which you can see here. The bond is broken down, so now the nitrogenous bases have unzipped, right? So once they unzip, there will be a formation of mRNA. There'll be one strand of DNA which is going to act as a template. So this strand will bond with the nitrogenous bases that are found moving around inside the nucleus, right? So the DNA strand is basically used to form a complementary strand of mRNA. So this is done by using the free nucleotides, as I said, which are found in the nucleoplasm. So the mRNA, which is this one here, will code the strand of DNA. So the strand of DNA will be this one, which has a code on how to manufacture a protein. Then this mRNA will create a code which matches to this DNA here. So once it is created, the code is received by means of what we call codons. So the codons are the three nitrogenous bases of the mRNA coding to the DNA, to each triplet of the DNA. So once the code is received from the DNA to the mRNA will move outside of the nucleus to the cytoplasm and straight to the ribosomes, which are found outside of the nucleus. And remembering that ribosomes are important in protein synthesis. So this stage, which is stage 1, this stage is called transcription. So this is the stage where the DNA unwinds with the help of enzymes, breaking the bond, creating an mRNA which takes the code from the DNA on how to manufacture proteins. Then the mRNA moves outside of the nucleus to the cytoplasm all the way to a ribosomes, which is responsible for the manufacture of proteins. So now we're going to focus on the process of translation. So what this means is the message that was carried by the mRNA from the first stage, which was the transcription stage, which was received from the DNA inside the nucleus, that message is taken over to the cytoplasm inside the ribosomes. So the first thing that's going to happen is the tRNA, which is this one here, has 3 nitrogenous bases, these ones here, which are known as anticodons. So these 3 are known as anticodons. So as you can see, they're trying to take or to match with the codons of the mRNA. So making them complementary, right? So which means the codons and the anticodons are complementary. According to the codons on the mRNA, the tRNA will bring the required amino acids to the ribosome. So what's going to happen is this tRNA, as it goes to the mRNA, it's taking the message on how to manufacture or to make proteins, and it's gonna take it all the way to ribosomes. So if these are ribosomes, it's going to take it to these ribosomes and show them on how to manufacture proteins using this code of amino acids. So the amino acids are actually linked by peptide bonds to form the required proteins. So as you can see here, this is a protein that is actually forming as a result of these tRNAs receiving the message or code from the mRNA. Thank you so much for watching. If you have any questions, suggestions, or comments, please write them in the comments section below. Don't forget to subscribe to the channel for more videos.